Hello and welcome to my next video on biotechnology. Now, biotechnology is technology that is based on biology and involves the exploitation of living organisms or biological processes to improve agriculture, animal husbandry, food science, medicine and industry. Now, examples you need to know, these are all the ones in the book, and yeah, you just need to know them. Cheese and yogurt making use the lactobacillus bacterium. That's naturally grows in milk, changes the flavour and texture of the milk to generate a different food. These bacteria prevent the growth of other bacteria that would cause spoilage and so preserves the food. So that's why cheese was made in the first place, was just a way of preserving milk. Mycoprotein, which is also known as corn, so a meat alternative for vegetarians. Now that's a growth of a fusarium fungus in a culture. The fungal mycelium produced is separated and processed as food. If you remember, a feature of fungi is that they can produce mycelium threads, and that's what mycoprotein is made of. Naturally brewed soya sauce. That's roasted soybeans are fermented with yeast or fungi such as aspergillus. Penicillin, an antibiotic medicine, we'll look at this a little bit more later. That's penicillium is grown in culture and produces the antibiotic penicillin as a byproduct of its metabolism, specifically a secondary metabolite, which we'll look at. Insulin. E. coli is generally modified to carry the human insulin gene. Again, this will be looked at in another video. Organisms secrete the insulin protein as they grow. Pectinase, which is used in fruit juice extraction. The Aspergillus niger is grown in certain conditions, produces and secretes pectinase enzyme. Calcium citrate, which is used in detergents. Aspergillus niger again produces citric acid as a byproduct of its normal metabolism. Biogas fuel production. Methanogenic bacteria grown on concentrated sewage respire anaerobically and generate gases that can be used as fuel. And wastewater treatment. A variety of bacteria and fungi use organic waste in the water as nutrients and make the waste harmless. For example, fusarium grown on corn steep liquor, a waste product of the corn milling industry. So there are just some examples. They're in your book. You can read over them, but you just need to know about them. So what are the advantages of using microorganisms? Why do we use them? Well, they can grow rapidly in favoured conditions, in their optimum conditions. Some bacteria can replicate every 20 minutes. Now, they can often produce proteins or chemicals that are given out into surrounding medium, so that means they can be harvested quite easily. They can be genetically engineered to produce specific products, with the example of insulin. They can grow well at relatively low temperatures, much lower than those required in chemical engineering or similar processes. So, for example, the Haber process, which produces ammonia, requires, I think, 500 degrees Celsius. Anything that uses microorganisms is generally done around body temperature of 30 degrees, give or take, depending on the bacterium. But you rarely find something that's more than 100 because it just boils, so very low temperatures, which keeps costs down. They can be grown anywhere in the world and are not dependent on climate. Since they're grown like fermenters, you can grow them anywhere. You don't need to grow them in a specific warm or cold climate. Tend to generate products that are in more pure form than they're generated via chemical processes. Because they have enzymes, they tend to make just one exact product. In chemistry, you can have equilibrium where if you're doing with ammonia, hydrogen, nitrogen becomes ammonia. You can get some ammonia, but you also stick with the original nitrogen, hydrogen. Here, you tend to get a full conversion. And also, if you're trying to make, let's say, a certain drug that has to be in a certain shape, enzymes will create that certain shape rather than a different shape of the same molecule. So you get, good pure chemicals. And they can often be grown using nutrient materials that would otherwise be toxic or useless to humans. For example, ethanol can be used as a food almost, or nutrients for bacteria, which is toxic to us. So we can get rid of waste products that we don't want. Now here is the growth curve. Now, this is the standard growth curve. Of course, it isn't always 100%, but this happens in a closed culture, but it happens in all over. It doesn't have to be just bacterium or microorganisms. It happens in proper big environments. So there are 4 phases. Number 1 is the lag phase. This is when organisms are adjusting to surrounding conditions. This may mean taking in water, cell expansion, activating specific genes, and synthesizing specific enzymes. The cells are active but not reproducing because they're growing. Think of them like, if you have a baby, it will still be taking in stuff but won't be quite ready to reproduce yet. The length of this period depends on the growing conditions. So the more stuff you have in there, the better the stuff you have in there, then the better and faster they will go out of the lag phase. You then got the log phase. You get the lag phase and then the log phase, also known as the exponential phase, because the population size doubles each generation. So think about it, if you have bacterium, 1 splits to 2, splits to 4, splits to 8, etc. And every individual has enough space and nutrients to reproduce. Now this can happen every 20 to 30 minutes. The length of this phase depends on how quickly the organisms reproduce and take up the available nutrients and space. So this stage continues as long as there is enough nutrients for each organism to survive and enough space for each organism to survive. Then you reach stage 3, the stationary phase. The nutrient levels decrease and waste products like carbon dioxide and other metabolites build up. Individual organisms die at the same rate at which new individuals are being produced. Now in an open system, like in the environment, this would be the carrying capacity, so it would stay roughly at this level. But in a closed system where you're not adding new nutrients in, so there's only a set amount, you get the next stage, which is the death stage. So basically at stage 3, it's when birth rate equals death rate, because there's nutrients are running out, space is running out. Now the decline or death phase. Nutrient exhaustion and increased levels of toxic waste products and metabolites lead to death rate increasing above the reproduction rate. Eventually all organisms will die in a closed system, where they've used up all their nutrients and they're now starving and they're producing toxic chemicals which are toxic to them. Which, for example, let's say they were producing sulfuric acid. Well, that's going to start burning them alive. So that's a closed system. An open system, you have 1 to 3. So that's, perhaps on the savannah with zebra. 1 to 3. Stage 4 comes about when you have a closed system. Now, metabolites. Now, metabolites are just the process of, the products of metabolism. Now, a metabolism refers to the sum total of all the chemical reactions that undergo the body. The waste products of some organic metabolic processes are the vital nutrients required by other organisms, so we will harvest these metabolites. And there are 2 types, primary metabolites and secondary metabolites. Primary metabolites are produced as part of the normal growth. They are needed by the organism. So, for example, amino acids, proteins, enzymes, nucleic acids, ethanol and lactate. So, as they will naturally, respire, they will naturally give off certain chemicals. For example, carbon dioxide will be naturally given off. Now, that's going to match the growth in the population size. As more of the population grows, more of these things will be given off. So, it kind of matches it. So, the dotted line is the number of primary metabolites. See, it's very close to the population size, but these processes have to occur for the organism to live. So you get these metabolites. If it is not producing primary metabolites, it will die. Secondary metabolites are substances produced by an organism that are not part of its normal growth. Now these are generally not needed for the organism to survive and are produced when you've got a kind of a good part after the growth period. Now, these are very often antibiotic chemicals, such as penicillin. And the production of secondary metabolites usually begins after the main growth period of the organisms, and so does not match the growth in population of the organism. So as you can see, it happens a lot later and ends up becoming a lot higher. Now, all organisms produce primary metabolites, only some produce secondary. So penicillin is a secondary metabolite, but mycoprotein is a primary because it's produced naturally by fungus or fungi. So without that process occurring, fungi would die. Fermenters. Now, I haven't drawn a picture of a fermenter here because it'll take forever, but you can find one online, and I'm sure you'll know what a fermenter looks like. So, I mean, fermenters, the things you need to remember is it has, like paddles for stirring, air inlet, water jacket to cool it, where there's an inlet and an outlet, inlet for the addition of nutrients, there's a motor which rotates things, loads of different probes for pH and temperature and oxygen, all these different things. And there are two types you need to know. The batch or paddle fermenter, which is kind of the classic fermenter with the paddles, and a loop fermenter, which is a continuous kind of just big loop. And they're known as many different names, but you can find them online. But the important thing is what the fermenters do. And they control conditions. So the conditions inside for the organisms or the microorganisms are perfect. So things that they will control is temperature. Too hot and enzymes will denature. Too cool and growth will be slowed. So you need optimum temperature. Type and time of addition of nutrients. Now, different microorganisms require certain nutrients. You have to give the right type, and you also need to make sure you add them, at correct times. Now, if you want primary metabolites or secondary metabolites, you will add the nutrients at different times. Oxygen concentration. Now, most applications of microorganisms require aerobic conditions, so you must have a lot of oxygen. Lack of oxygen would lead to unwanted products of anaerobic respiration. For example, if you use bacteria, you could get ethanol, which you wouldn't want in this case. But equally, you might want anaerobic conditions because some organisms produce the correct materials in anaerobic conditions. So it can also, make sure you've got, if the oxygen concentration you need is 0, it makes sure you have that. And pH. Changes in pH will affect the activity of enzymes and reduce growth rate. Now, two types of cultures. There's a batch culture and a continuous culture. Now, a batch culture is when microorganisms are put into a fermenter with a set amount of nutrients. It's all been pre-planned and put in. They're allowed to grow for a fixed period of time. No further nutrients are added. And at the end, the products are removed and the fermentation tank is emptied. An example of this is penicillin. A continuous culture is where nutrients are added all the time. And the products are removed all the time. So it can happen at regular intervals, but generally could happen continuously. And hormones such as insulin or mycoprotein, they all use these sorts. So, the benefits of a batch culture, or rather the features of a batch culture, not all benefits. The growth rate is quite slow because the nutrient level declines with time. So if you have, enough food for all the organisms and it starts decreasing, it's going to cause the growth rate to slow down as opposed to having always enough nutrients. It's easy to set up and maintain, relatively. We can do it, but trained scientists can. Or, trained people in fermenters can. If contamination occurs, you lose one batch. You'll just have to get rid of the whole batch and start again. Clean it, done. Less efficient. Fermenters, since fermenters is not in use all the time, you put some stuff in, wait, take it out, sterilize, put it back in, take it out. You're not getting as much of a product as you could. And this is generally used for secondary metabolites. Continuous culture, it's much faster because you're getting continuous nutrients. It's more difficult, and the maintenance of required growing conditions can be difficult to achieve, so it's harder to get the correct conditions you want. If contamination occurs, huge volumes of product may be lost because it's a continuous culture. So you have to completely take out everything and restart the whole thing. And basically any product which has been in there during the session where it got contaminated. So if you had a whole, been doing it for a whole day and suddenly get contamination, you can't use the stuff you made for the whole day. As opposed to a batch culture, which, it might take, an hour to do it, and you just lost that hour's work. It wouldn't be that short amount of time, but it's just an example. It's more efficient because it's always on. And it's very good at producing primary metabolites, such as insulin and such as mycoprotein. Asepsis. Asepsis is the absence of unwanted microorganisms. You do not want contamination. And so you use aseptic techniques. And this refers to any measure taken at any point in a biotechnological process to ensure that unwanted microorganisms do not contaminate the culture that is being grown or the products that are extracted. Now, why we don't want unwanted microorganisms? Well, why wouldn't we want them? Because they're unwanted. But the reason why is because they compete with the culture microorganisms for nutrients and space. This will reduce the yield of useful products. It may cause spoilage, may produce toxic chemicals, or may even kill the cultured microorganisms and their products. So we want asepsis. And there are two ways you can use aseptic techniques that we need to know about. That's if you're doing it at a laboratory level or if you're doing it at a large scale level. So, at a laboratory level, you have sterilisation, of course. Now that can be with heat or with UV light, steam, or an autoclave. It's like a large pressure cooker, just to get rid of any bacteria. Now work can be carried out in a fume cupboard. Now this means that air circulation carries any airborne contaminant away from the bench space because the air rises. And cultures of microorganisms are kept closed where possible and away from a bench surface when open in use. So let's say you're doing an agar plate, for example. You want that closed for the least amount of time to prevent bacteria getting in. On a large scale, you do cleaning, basically washing, disinfecting, steam cleaning the fermenter and associated pipes when not in use, removes excess nutrient medium and kills microorganisms. Stainless steel is generally used to make the surfaces of fermenters to prevent microbes and medium sticking to the surfaces. Again, sterilising all nutrient media before adding to the fermenter. And fine filters on the inlet and outlet pipes avoid microorganisms entering or leaving the fermentation vessel. Pretty standard. Yeah, makes sense. Now, enzymes. Now, we all know that enzymes are catalysts. They increase the rate of reaction. Without them, it would take forever to do anything in biology. Now, enzymes in biology can be used for quite a lot of things. Now, why they are good as opposed to chemical processes is that they can work at particularly low temperatures, as we said earlier. And basically, it's more efficient to just isolate the enzyme and use just the enzyme rather than the whole organism to get that one specific desired product. So if you use the enzyme, you're guaranteed to get just the one product the enzyme makes. If you use a microorganism, you get loads of different products as well, because no organism produces just one thing. Now, isolated enzymes can be produced in large quantities in commercial biotechnological processes. The extraction of enzymes from the fermentation mixture is known as downstreaming. And this is a process involving the separation and purification of any product of a large-scale fermentation. Now, we want to immobilise the enzymes, and that is basically separating them from a mixture. And there are four main processes for immobilising enzymes you need to know about. And the reason why you choose ones over another might be preparation cost, relative importance of enzyme leakage, and efficiency. Now, adsorption, that's with a D, not a B, as chemists will know. That is when a porous support such as clay binds to enzyme molecules non-covalently. So they mix with the immobilising support and bind to it due to a combination of hydrophobic interactions and ionic links. So ionic is between positive and negative, hydrophobic is water hating. So for example, with phospholipids, you have the heads which are hydrophilic facing outwards and the tails facing inwards due to just, that's kind of like hydrophobic interactions. Now, I will come to the disadvantages of them in a second of each one, but we'll just say what they are. Covalent bonding is exactly the same except with covalent bonds. It has to be an insoluble material such as clay. And also, if you use cross-linking agents, such as some examples that are in the book, that's glutaraldehyde or cephorose, you can then get the enzymes forming covalent bonds with each other, as you can see, kind of forming a chain. Entrapment. Enzymes may be trapped, for example, in a gel bead or a network of cellulose fibres. The enzymes are trapped in their natural state, and you kind of got like a little prison for them. And membrane separation. You will have a partially permeable membrane and two sections. One section will have the enzyme solution, the other will be where the rest of the solution kind of comes through. And then subject molecules can pass through, and then the products can pass back, but the enzymes are too big to pass through. Now, the disadvantages of adsorption. The bonding forces are not particularly strong. Enzymes can become detached, and this is known as leakage. However, provided the enzyme molecules are held so that the active site is not changed and is displayed, absorption can give very high reaction rates. Basically, as you can see here, all 4 active sites are open. They are easily gettable. You can just, the molecule can get to it quite easy. So you get very quick reaction rates because lots of reactions can occur. In covalent bonding, it does not immobilize a large quantity of enzymes, but the binding is very strong, so there's little leakage. So adsorption, you can get a lot of things bonded together, but leakage could lower the yield from that. Covalent bonding, you get less things bonded together, but the yield of that is a lot higher. So it's kind of, they both have benefits. Entrapment. Reaction rates can be reduced because substrate molecules need to get through the trapping barrier, so the cellulose gaps. And that means the active site is less easily available, so you can reduce reaction rate there just because they can't reach it. And membrane separation, product molecule, the only problem is you might not get all the substrate molecules, all the product molecules going through that partially permeable membrane. So that could be a problem. So advantages and disadvantages of immobilizing enzymes. Advantages. Enzymes are not present with products, so purification and downstreaming processes costs are low. Now, downstreaming is when you're separating, purifying of the product. So with enzymes, you only get the products remained. Well, you get the enzymes and the products together, so it's much easier to separate them. Remember, and it's 100% pure. You literally will have just product, maybe a few bits of reactant left, but still, 3 things in a mixture isn't that hard to separate, as opposed to using an organism where you could have, hundreds of chemicals in there. The enzymes are immediately available for reuse. Enzymes don't get used up, so you can use them straight again. It's particularly useful in allowing for continuous processes. And immobilized enzymes are more stable because the immobilizing matrix protects the enzyme molecule. So basically the enzyme won't get denatured as easily, won't get destroyed. Disadvantages, however, immobilization requires additional time, equipment and materials, and so is more expensive. Immobilized enzymes can be less active because they do not mix freely with substrate. There's a very set way they must be, put together. So it's not enzymes and substrate all mixed together. It's enzymes in one place, substrate in another. And any contamination is costly to deal with because the whole system would need to be stopped. It's a bit like kind of continuous processes. And that is that. So conclusion, biotechnology is basically using microorganisms and biological processes to help technology. You need to know the standard growth curve of an open and a closed system. Fermenters, know how it works, know about batch cultures and continuous cultures. Asepsis is the removal of unwanted microorganisms. And you need to know about the immobilization of enzymes. That's just to trap them so we can use them on their own. So thank you for watching. As usual, if you have any questions, ask, comment, like, email, subscribe, the usual. It's getting reasonably close to exams now. For AS, it's a month and a bit away. A2, we've got 2 and a bit months. So really, this is the time to ask questions now, because when it gets to near the exams, I'm going to be quite busy doing my own exams. While I will try and help, it's going to be quite hard to, so get revision done now, and any problems, ask now. Thank you for watching, and goodbye.